Good morning. Let's discuss on folic acid. Folic acid or vitamin B9 is a very important vitamin crucial for proper brain functions and plays an important role in mental and emotional health. So structurally, folic acid consists of three components. One is the pteridine ring, one is the para-aminobenzoic acid or PABA, one is the L-glutamic acid. So this is the structure. This is the pteridine derivative, then PABA, para-aminobenzoic acid, and this is the glutamic acid. Pteridine derivative and PABA combinedly they form the pteroic acid, and once they combine with the glutamic acid, they form the complete structure of folic acid. Active form of folic acid is tetrahydrofolate, and sources the folic acid is found in green leafy vegetables, liver, yeast. The word folate is being derived from the Latin word folium, which means leaf. The nutritional requirement is 200 milligrams per day, and requirement increases during the pregnancy and lactations. This is the structure of folic acid, how it is being converted up into the active form, that is a tetrahydrofolate. So folate reductase is an enzyme which leads to the reduction of this folic acid ring in the presence of vitamin C and NADPH. So as a source of hydrogen. So these 5 and 6 position double bond is being reduced, and this converts up into the dihydrofolate. Further, this dihydrofolate with the help of same enzyme can be converted up into the tetrahydrofolate by reduction of at another double bond which is at 8 position. Function wise, the tetrahydrofolate acts as a carrier of one carbon unit. The one carbon units are formyl group, formamino groups, methynyl group, methylene group, hydroxymethyl groups, and methyl groups. That's how it is very much involved in the one carbon metabolism. And tetrahydrofolate coenzymes serve as an acceptor or donor of one carbon units in a variety of reactions involved in amino acids and nucleic acid metabolism. The methyl group in N5 methyl tetrahydrofolate is used for synthesis of active methionine, which takes parts in transmethylation reaction. Already we have discussed when I talked on methionine metabolism. Such transmethylation reactions are required for synthesis of choline, epinephrine, and creatine, etc. So these are the two sites, N5 and N10 position. These are act as in the site where the one carbon species can be loaded. So in the loaded state, the one carbon species can be, carried to the various site in various metabolic reaction. Wherever it is being required, it is donated to those sites. You can see it here, the N5 and N10, they can easily load the one carbon species. Then the 5 major reactions in which the tetrahydrofolate is involved. One is conversion of serine to glycine. Only difference in glycine to and serine is that is of CH2OH group. So conversion of serine to glycine is accompanied by the formation of N5 and 10 methylene tetrahydrofolate. So this methylene group is taken up from the serine, and that's how serine is being converted up into the glycine. Similarly, the synthesis of thymidylate, that is a pyrimidine nucleotide. The enzyme thymidylate synthase that converts the deoxyuridylate into the thymidylate uses N5 N10 methylene tetrahydrofolate as a methyl donor for this reaction. So that's how the thymidylate or the thymidylate monophosphate is required for synthesis of DNA. This reaction becomes very, very important because it becomes the source of these nucleotide for synthesis of DNA. Then catabolism of histidine. Histidine in the course of its catabolism, you will see in subsequent slide how it is being converted up into the glutamate. So in course of its conversion, it is converted up into the formamino-glutamate, and this molecules donates the formamino group to the tetrahydrofolate to produce the N5 formamino tetrahydrofolate. Then the fourth reaction is the synthesis of purine. So N5-formyl tetrahydrofolate act as an intermediate formed in the histidine catabolism and is used in the biosynthesis of purine, and therefore it is used in the formation of DNAs and RNAs. So without the presence of folic acid, we cannot expect these reactions, and they mostly affect the DNA and RNA synthesis. Then synthesis of methionine from homocysteine. Only difference between the methionine and homocysteine is of one methyl group. So homocysteine is converted up into the methionine in the presence of N5-methyl tetrafolate and vitamin B12. So this methyl group is being loaded onto the tetrahydrofolate, which acts as a donor and gives homocysteine, and that's how homocysteine is converted to methionine. Vitamin B12 acts as a facilitator. So there is a more clear vision of all these reactions, how the homocysteine is converting up homocysteine to methionine. So methionine synthase is the enzyme. Cobalamin or vitamin B12 is converted up into the methylcobalamin, which itself is converted up via the donation of methyl group from tetrahydrofolate. So this is methyl tetrahydrofolate in turn gives methyl group to cobalamin, and it converts up into the tetrahydrofolate. This is an irreversible reaction, and this is the only reaction which frees the methyl group. So in case of deficiency of vitamin B12, this methyl remain trapped to the tetrahydrofolate, and this lead to the severe deficiency of tetrahydrofolate, which is being very much required in various metabolic reaction for one carbon purposes. So this condition is known as folate trap. So cobalamin to methylcobalamin, and methylcobalamin again converts up into the cobalamin, and this releases its methyl group to homocysteine, and that's how homocysteine is converting to methionine. Very, very important reaction. So this is a nutshell, the whole view of all the reaction in which the tetrahydrofolate participate. This is the dietary folic acid, which converts up into the dihydrofolate with the help of this enzyme, folate reductase and vitamin C. Then dihydrofolate to tetrahydrofolate, and how it is being utilized? Tetrahydrofolate to methylene tetrahydrofolate with the help of vitamin B6 or PLP. Here serine is converting up into the glycine. Then further methylene tetrahydrofolate is utilized for synthesis of this thymidylate monophosphate. So this uridine monophosphate is converted up into the thymidylate monophosphate, which is very much important for synthesis of DNA. If this reaction is hampered due to the deficiency of vitamin B12 or due to the deficiency of this tetrahydrofolate, so this will worstly affect the synthesis of DNA. So in course of this conversion, then tetrahydrofolate converts back into the dihydrofolate, which can again converts to tetrahydrofolate. Further, tetrahydrofolate can convert up into the formyl tetrahydrofolate. Tetrahydrofolate. Histidine in course of its metabolism is converted up into figlu or pharmaminoglutamate, and pharmaminoglutamate further converts up into the glutamate with the help of folate. So if there is a deficiency of folate or folic acid, there is no conversion of figlu to L-glutamate, and it remain in the blood, and further it is excreted through urine. Further, this formyl tetrahydrofolate can be utilized for synthesis of C2 and C8 carbons of purine. So that's how it is very, very important for synthesis of purine nucleus. So you can see the versatile role of tetrahydrofolate, especially the synthesis of DNAs and RNAs, which are dependent on purines, plus thymidylate monophosphate, which is an important nucleotide for synthesis of DNA. Then methyl tetrahydrofolate, I have already told you about the methyl trap. It is entrapped with tetrahydrofolate, and which is to be released with the help of vitamin B12. And here homocysteine is act as an acceptor of methyl group, and which converts up into the methionine. The enzyme is methionine synthase. If there is an enzyme deficiency defect or there is a vitamin B12 deficiency, it will lead to homocysteinemia, because Homocysteine is not able to be converted to methionine, and methyl gets entrapped permanently into the tetrahydrofolate. This lead to the severe deficiency of tetrahydrofolate as well. So this is folate trap. Then tetrahydrofolate. So this is playing a pivotal role in various metabolic reactions, especially of amino acids and of nucleic acid metabolism. Then this step 1 explains the folate trap. Step 2 is important for methyl transfer reaction. The same reaction, you can see it here, how the conversion of the one carbon pool is here. So methyl tetrahydrofolic acid, it converted up into the tetrahydrofolate, which leads to conversion of vitamin B12 to methylcobalamin, which is important for conversion of homocysteine into methionine. You have seen in the previous slide also. This methionine is converted up into the S-adenosyl methionine, which is the active form of methionine. With the help of ATP, adenosyl group is being added here. And then S-adenosyl methionine again converted up into the S-adenosyl homocysteine. With the help of release of one methyl group, with the help of enzyme methyl transferase, this methyl group is being taken up by methyl acceptor in the form of various intermediates like creatine and then epinephrine, etc. are there. So methyl acceptor can be converted it up into the methylated product. Then what are the inhibitors of this folic acid? Sulfonamide is the major inhibitor because structurally they are very quite similar to the folic acid. It acts as a competitive inhibitor of PABA, para-aminobenzoic acid. Is structurally very similar. This portion is very similar to the sulfonamide. This inhibits the folic acid synthesis, and that's how inhibits the bacterial growth. So next is aminopterin and amitriptyline. Methotrexate, that is an anti-cancer drug. So competitive inhibitor of dihydrofolate reductase, and which also inhibits the tetrahydrofolate. Then it inhibits the cell division and leading to the death of cancer cells. This is the mechanism of how aminopterin and amitriptyline work onto the cancer cell. Then perimethenamide, that is an anti-malarial drug, causes the which leads to the severe folate deficiency. What are the causes of deficiency? Pregnancy, anticonvulsant drug like phenobarbital, hydantoin. Then dietary deficiency will also lead to folic acid deficiency, defective absorption also like intestinal bowel syndrome. Then celiac sprue, gluten-induced enteropathy. All these conditions lead to the severe folic acid deficiency. GI surgery, gastrointestinal surgeries, jejunomectomy or gastroileostomy. Then folate trap due to the B12 deficiency and homolytic amine. All these conditions lead to severe folic acid deficiency. So deficiency manifestations are in the form of megaloblastic or macrocytic anemia, because you have seen the role of folic acid in synthesis of thymidylate monophosphate, which is an important nucleotide for synthesis of DNAs, RNAs. And so due to the deficiency of this folic acid, purine ring is also not being formed. So DNA RNA synthesis is worstly affected. Cell cycle is arrested in S phase, and cell grows without the cytoplasm. So it is a very peculiar in structure, which is known as megaloblastic or microcytic RBCs. Accumulation and excretion of FIGLU in the urine is there, which is an intermediate of histidine metabolism. So FIGLU is not able to be converted to glutamic acid. It is trapped in this form and it is excreted in the urine. Then hyperhomocystinemia is there. Then neural tube defect in fetus in the form of spina bifida and anencephaly. Then AICR excretion. AICR is actually amino imidazole carboxamide ribose 5-phosphate, which is actually addition of C2 with the help of this N10 formyl THFA in the last step of purine synthesis. When this reaction is blocked, the precursor in the form of AICAR, that is amino-imidazole carboxamide ribose 5-phosphate accumulates and is excreted as such in the urine. Live diagnosis can be done on the basis of peripheral smear and macrocytosis and reticulocytosis, which is the abnormal number of immature RBCs. Then histidine, second is the histidine load test and excretion of figlu in urine, because figlu is not able to be converted into the glutamate, L-glutamate, in the absence of this tetrahydrofolate. So it starts coming in the urine. Histidine metabolism is affected in the deficiency. This is an indicator of folate deficiency. Then serum folate level estimation is another way in order to diagnose about the deficiency of folic acid. Thank you. There is a case. A 30-year-old pregnant lady came to the OPD with complaint of breathlessness, weakness, loss of weight, and smoothness of tongue. On lab investigation, the peripheral smear shows macrocytic RBCs and reticulocytosis. So you can better understand how it happens and when it happens. Hemoglobin 6 milligrams per DL. Then urine was positive for figlu, formamino-glutamate, USG finding suggestive of neural tube defect in fetus. So what is your probable diagnosis, and mention the deficient compound so that you can easily crack this case, and this clearly shows the deficiency of folic acid.